All right, good afternoon, ladies and gentlemen. We are going to go ahead, oops, there we go, and talk about enzymes today. So this is in our unit 3 that's called Cellular Energetics, which includes enzymes, cell respiration, and photosynthesis. So today we're going to talk about enzymes, and this is mostly found in chapter 6. All right, so big questions that we're going to be looking for. How do living systems control their metabolism? So metabolism refers to all the chemical reactions that are happening inside of your body, and enzymes are involved in every single one of those. Okay, maybe exaggerated a little bit, but pretty much every single reaction is being controlled by enzymes. And enzymes are incredibly fast at making reactions occur, and they can be used over and over and over again. So a reaction can happen incredibly fast. So at some point, your body wants to stop the reaction from happening. It wants to be able to control the reaction. And so that's what we're going to be looking at today. And then how do living systems carry out a wide variety of specific chemical reactions? And that has to do with how specific each enzyme is. All right, so let's go ahead and take a look. First of all, oops, my picture's in the way on this one. Enzymes almost always in biology end with the letters ASE. It's a common nomenclature suffix for enzymes. So we're going to do a liver lab if you haven't done it already. That's the enzyme is called catalase. There's topoisomerase. There are so many ACEs. I'm having trouble thinking of all of them right now. And I'd like you to take a look at the diagram. In AP biology, the graphs and diagrams are so incredibly important. So let's take a look and kind of pull apart this graph, because I can guarantee you that I will put this on a test, and I can almost guarantee you that you'll see this on the AP exam. So take a look along the bottom axis. It says progress of the reaction. So the reaction, whether it's moving forward or not. Along the side, it says free energy. So we're talking about how much energy is required to get this reaction to progress, to move forward. So if it's a normal reaction and there's no enzyme involved, and that's the black line that's in the graph here, you see that it requires lots of input of energy in order for that reaction to then move forward. So we have to add a lot of energy. When there's an enzyme present, we don't need to add anywhere near as much energy, and the reaction progresses anyway. So let me give you like a really easy example of this. In the liver lab, we're going to be, if you haven't done it already, in the liver lab, we're going to be looking at the decomposition of hydrogen peroxide. Hydrogen peroxide breaks into water and oxygen. And that reaction can happen naturally. However, you have enzymes in your body that make it happen incredibly fast. Well, how does that reaction happen naturally? There's only a few ways. We have to add energy to the system. So let's just imagine, maybe some of you have seen bottles of hydrogen peroxide that you can get at like the pharmacy or something like that. Hydrogen peroxide is good for disinfecting cuts. It touches the catalase in your blood, and then that turns the hydrogen peroxide into water, and oxygen bubbles, so it bubbles on your cut, and that washes your wound, and it introduces oxygen, which is deadly to most anaerobic bacteria, which are the bacteria we're usually afraid of getting infections from. So that's how it works. So that's that. Imagine we have one of those bottles. It has a shelf life. It can only last for so long before it eventually decomposes. How could we speed up the rate of decomposition? Well, if we took that bottle and we poured it into a dish and set it out in the sun, the sun would add energy, and it would, over time, cause the decomposition of the hydrogen peroxide. So that's why hydrogen peroxide bottles are dark brown, so that the energy from the sun can't cause that reaction to speed it up. We don't want it. We want it to have the longest shelf life possible. We could also take that bottle of hydrogen peroxide and pour it into an oven. What am I trying to say? Oh my gosh, a frying pan. No. A pot. That's the word. I couldn't think of the word pot, everybody. We want to pour it into a pot and put it on the stove and boil it. We just added a ton of energy. And when we add a ton of energy, we're going to speed up that decomposition. We're going to cause it to break into water and oxygen. We could even take that brown bottle and shake it. Just shake it and shake it and shake it. And that's adding energy. So these examples that I'm giving you, those are all the black line. That's how we would add energy. And technically, if we just leave it on the shelf for long periods of time, there's enough energy that gets added to that system incredibly slowly that eventually that hydrogen peroxide is going to decompose into water and oxygen. In your body, we can change that hydrogen peroxide into water and oxygen in I'll make up a number because I don't know the number, a billionth of a second. The reaction happens super fast, and that's because you have enzymes that are specifically for breaking up hydrogen peroxide. Okay, so how do these enzymes work? Now I've given you some examples here. So the general idea, let's get my picture out of the way. So enzymes interact with their reactants, and super important that you learn the word substrate. So substrate is the molecule that we're working on. So in my example, the substrate is the hydrogen peroxide. So hydrogen peroxide is going to, let's say we've got the reactants here. This is not a perfect example because this is where the enzyme is changing to reactants. Doesn't matter. Let's just imagine that hydrogen peroxide is right here. It fits in perfectly to this little spot on the enzyme substrate complex, kind of like a puzzle piece, only the puzzle pieces are bendy, which we'll talk about in just a second. So the enzyme is going to either form bonds or it's going to break bonds. And in the case of hydrogen peroxide, the enzyme that's called catalase breaks bonds and causes this water and oxygen to form from the hydrogen peroxide. And then those would be the products right there. And now the enzyme returns back to its shape and it's ready to do it again. And it can repeat over and over and over again. And the enzyme never gets used up in the reaction. So another important vocabulary word when we're dealing with enzymes is the idea of the active site. So the active site is the area of the enzyme where the molecule, where the substrate fits. And again, it bends a little bit. You can see the shape change just a little bit, and try to make really put all this together. We've talked about proteins denaturing. So enzymes are a type of protein. Their shape is incredibly important because it has to fit the molecule that it's causing this reaction to happen in. And so if we have a mutation in our DNA that causes the wrong amino acid to join, that changes the tertiary structure, trying to pull it all together, that changes the tertiary structure of the protein, of the enzyme. So now it's the wrong shape. So now my reactants won't fit and none of that reaction is going to happen. And then that leads to some sort of the symptoms of a genetic disorder. Okay, so moving on, let's look at the details a little bit more. So instead of it being a total puzzle piece, we're going to call this the induced fit model. And induced fit means it's more like a bendy puzzle piece. So the shape of the active site of an enzyme is shape specific for a particular substrate. The binding of a substrate to the active site induces necessary, well, it gets wordy here, conformational changes. So the enzyme changes its conformation, its shape. So conformational change of the enzyme to catalyze the reaction. And to catalyze the reaction means to speed up the reaction. So as it fits in, the enzyme changes, speeds up the reaction, and then it returns back to its original shape once the substrate or the products have left. All right, and this is just showing you an example. So notice the shape of the enzyme. Notice that the enzyme has bent a little bit while the substrate is inside of it. All right, moving on. These are just a couple of examples of enzymes. There are so many enzymes that we're going to stumble across. Topoisomerase is one that we'll come across in our DNA unit. It's involved in limiting stress on DNA during replication when the DNA is getting copied. And we'll talk about that a little bit later, topoisomerase. And then Rubisco is coming up in this unit. Get my picture out of the way. Rubisco is coming up in this unit. It's important during photosynthesis where the plant is pulling, usually a plant, the chloroplast, is pulling carbon dioxide out of the atmosphere and then it's attaching it to another molecule to make sugar. Rubisco is absolutely key to all that. All right, so then we're going to talk about cofactors. Most enzymes require necessary compounds that you guys have all heard of, but you never knew what they were for: vitamins and minerals. And this is why it's important to make sure that we have well-balanced diets. If we're missing out on vitamins and minerals, then our enzymes sometimes aren't as efficient or can't do some of our reactions. So here's there are two good examples here. Magnesium and manganese. Those are two elements that are not usually in amino acids, but yet they're present in these super important. So rubisco is on the left, and then topoisomerase is on the right. And it's got these two ions that are present in that protein. They are super, super important with making the active site in that particular case super attractive to the molecule that it's trying to attract. So those are the vitamins and minerals, and one example. There are other ways that vitamins and minerals are important, but all right. This particular slide I'm not that into. Yeah, enzymes are super important. We can see sort of evolution of species as we see how their enzymes are different. We can look back at their DNA and we can see tiny little changes in their DNA that have led to different enzymes. And yeah, I'm not going to worry about that yet. We'll talk plenty about evolution in our evolution unit. Okay, so I mentioned earlier that these enzymes speed up these reactions. I think they usually say 1 billion times faster than the reaction would occur under normal circumstances. But regardless, at some point, the cell has to get some control. We don't want everything to happen all at one time. Sometimes we need a few of these reactions to occur. Sometimes we need a lot of them. Sometimes we don't want them to occur at all. So the cell has to regulate them. So we can either stimulate the reaction or we can inhibit the reaction. So in this particular slide, we're looking at competitive interactions and non-competitive interactions. So take a look at the diagram. I think it's really self-explanatory. A shows you the normal substrate fitting into the enzyme. That's the reaction that is supposed to be occurring. Now, if we don't want as much of that reaction occurring, there's two things. Well, there's many, but we're going to talk about two right now. We could have a competitive inhibitor. That would be another molecule that can fit in the active site, and it gets in the way of the substrate. So that in essence slows the process down. So instead of us digesting that molecule or breaking up that molecule or building that molecule incredibly fast, we're doing it a little bit slowly. We have competitive inhibition, so we've slowed it down. Non-competitive inhibition is the same idea, but instead of the non-competitive inhibitor, this other molecule, getting in the way of the active site, it actually touches the enzyme somewhere else. Now, I want you to think back about what you know about proteins. We had an FRQ question, for example, where what if we put a protein that's normally in water, what if we moved it into oil where it's a hydrophobic solution? And a lot of you told me that the protein would totally denature, it would change its shape so that the hydrophilic groups would go from being on the outside to the inside, etc. So you understand the impact of changing the biochemistry of the outside of a protein. So here we're adding a new molecule, and that new molecule is going to affect the R groups that are in that area, and it's going to change the conformation of the molecule. So if we have a non-competitive inhibitor, it caused the active site to change shape. It's no longer the right shape. Study the diagram, make sure you see that. It's no longer the right shape for fitting that substrate. And so we have inhibited that particular reaction. So again, competitive means it actively got in the way. Non-competitive means it changed the shape of the active site of the enzyme so the substrate couldn't fit in. All right, there's all kinds of other things that we can talk about. Oh, I didn't realize it was going to get close here. There we go. Allosteric interactions. So the word allosteric means other site. And if you can remember that, that'll actually help you a little bit because these words kind of will start spinning in your head, I know from experience. Allosteric interactions can stimulate or inhibit. So that means increase the reaction or decrease the reaction by causing a shape change in the enzyme. Everything about proteins is about their shapes. Don't ever forget that. anything that changes the shape of a protein, whether it's a mutation, whether it's the wrong amino acid because of a mutation, whether it's temperature, whether it's pH, whether it's the wrong solution, all kinds of things. An addition of a molecule, all those things change the shape of the protein, and they're going to affect how well that protein works. All right, so in the case of an allosteric inhibition, take a look at the diagram. Let's see if this one, it does go close up. Okay, take a look at the left side of the diagram where it says active, and then there's two arrows going up and down, and then it says inactive. So this enzyme oscillates. That means it goes back and forth between those two shapes under normal circumstances. So it does its work, and every time that it gets activated, it's activated when it's in the active form, and then it oscillates into the inactive form, and it oscillates back into the active form. It is possible for a molecule to lock in. Notice that it did not join in the active site, not in the little, this area. It didn't join there. It joined somewhere else, so other site. It joined, and it caused the active sites to all activate and stick that way. So that activator made the enzyme active, and it's not going to oscillate between its active and inactive form anymore. And vice versa, you could have an inhibitor stick into an other site, not the active site, not the site where the substrate is supposed to attach, but somewhere else on the enzyme. And notice that all of the active sites have now shut down, and it's permanently, I say permanently, until the inhibitor goes away, it's permanently in the off position. It is inactivated. So we've turned off that enzyme. If we don't want that enzyme to do its job, we need to turn it off, and that means inhibited. All right, moving on. Trying to see if there's anything I didn't say. It looks good to me. All right, let's get me out of the way again. Binding of a substrate molecule onto an active subunit of an enzyme can also trigger stabilization of the active conformation in all subunits. So if the substrate is present in, so the substrate, remember, is the molecule that we're trying to have the enzyme work on. In the example of our liver lab, it was the hydrogen peroxide. That's the molecule that we're trying to break up. If it's present and it attaches onto the active site of one enzyme, it causes all of the other subunits of that enzyme. Remember, that's the quaternary structure of a protein, a whole bunch of subunits. The presence of one causes the activation of all of them. Why? Because it means that you're in the presence of that molecule, and the cell wants to break up that molecule as efficiently as possible. So, for example, hydrogen peroxide is a poison inside of our cells. So if hydrogen peroxide were present, this is an analogy. This is not exactly the same. I'm just giving you an analogy. If we dropped this enzyme into a solution of hydrogen peroxide, this particular example enzyme, all of its active sites would lock into their correct conformation, and it would be able to react 4 times faster because it's all 4 times. I made that up, but faster because they're all locked in their correct or active conformation. It's not going to oscillate between that inactive and active form. Hope that made sense. So I think that sometimes students get mixed up between an activator, which is this molecule. In this case, the activator, it's not part of the reaction. It's not the substrate. It's not the hydrogen peroxide. It's some other molecule that tells that enzyme, hey, there's going to be hydrogen peroxide in the area. So an activator is not the same. And then if we go over here, this is actually the hydrogen peroxide. The hydrogen peroxide touches, it activates that particular enzyme, and then it stabilizes the 3 other subunits so that they're all ready to interact with any other hydrogen peroxide molecules that are in the area. So that's called cooperativity, when one subunit gets activated and the 3 other subunits then stabilize in their active conformation. All right, examples. So this is an example of an experiment that was done, get me out of the way, with an enzyme that's called caspase. I say capsase every single time, and that's wrong. The S comes before the P, so forgive me if I do stumble over it. Caspase. It's an important enzyme for apoptosis and necrosis. So apoptosis, remember, is programmed cell death. It's a normal, healthy, what your cells are supposed to do if something's wrong with them. Necrosis is cell death from something bad. So, for example, an infection, exposure to a toxin or a poison, something's happened and it's not supposed to, it wasn't programmed, it is dying because something else happened. That's called necrosis. But regardless, caspase is involved in helping the cell to die correctly. So this is an experiment just showing you what the scientists have been able to see. So they were trying to determine if caspase enzymes have an allosteric site. So remember, allosteric means other site. So that means, trying to help you remember this, that means that a molecule, not the substrate, not the hydrogen peroxide in my example, but some other molecule touches it and causes the shape to change. So that's what they want to know in this experiment. Does caspase have an allosteric site? So the active and inactive forms of caspase were already known. So that's this here. So there's the active form, and here's the inactive form. And so it says the allosteric, this is their hypothesis, allosteric inhibition of caspase 1 will lock the enzyme into an inactive conformation. So they add an allosteric inhibitor, and then it locks into a conformation that is too small. That's their hypothesis, that it's too small for the substrate that's supposed to fit in there. So notice the width of the pie piece here. You really need to study these diagrams because this is exactly what AP gives you on tests all the time, and you need to not glance over them. Study them, see if you can figure out what's going on. So notice that the pie piece is too skinny now, so the substrate, hydrogen peroxide, for example, can't fit in there. So now the results, this is the actual computer, there's a word for it and I'm suddenly proteomic, I don't remember the exact word, but the computer simulation of the shape of the molecule. And so they're showing you caspase, hard to say, caspase in its active form. They have allosterically inhibited, so they've added an inhibitor, and now it has changed shape. So they were able to look at that with a computer simulation and see it actually changing shape. And so that was part of the experiment. All right, moving on, a couple other things and then we're done. Compartmentalization, we talked about in our cells unit, how important it is. The organelles give us places for different reactions to occur in different spaces. And a lot of those reactions, pretty much all of those reactions, are being controlled by enzymes. And so you have different enzymes in different organelles. So the example here is the mitochondria. They say, just reading the diagram here, the matrix, remember that's the fluid inside of the mitochondria, contains enzymes in solution that are involved in one stage of cell respiration, but enzymes of a different stage of cell respiration, we're about to learn that this unit, are embedded in the inner membrane. So there's different locations for those enzymes so that the different reactions can happen in the different places. A real obvious example too would be like the lysosomes. They have certain enzymes in them that are separated from the rest of the cell, the hydrolytic enzymes that can cause apoptosis and digest food and that kind of thing. All right, super important, and this comes up in the liver lab. So whether you've done the liver lab or not, you just need to be aware. These are, again, study these graphs and make sure you get what's being said in these graphs. So I'll just read down below and then we'll look at the graphs together. So like all proteins, enzyme structure and therefore function, remember its structure determines its function. Anything that changes the structure completely changes its function. For the better or the worse, it changes its function. So can be affected by the conditions of the enzyme's environment. We already mentioned a couple of them. For example, that FRQ question where we moved a protein from a hydrophilic or water solution into a hydrophobic solution, and we said the enzyme would denature, it would change its shape because the R groups would swing to other positions. Same idea here. So if we change the environment in some way, it's going to impact those R groups, and they're going to change how they're bonding and how they're bonding with each other and how they're bonding with the environment. All right, there are three major environmental conditions that affect enzyme structure and function. We'll get to those in just a second. So let's take a look at these first two graphs. So along the bottom axis, I see temperature on the first graph, and it's talking about the optimal temperature for two enzymes. So notice that, let's look at the black enzyme. In this particular one, for a human enzyme, I was trying to see which enzyme it was. For a human enzyme versus a bacterial enzyme, which is thermophilic. Thermo means heat and phil means love. So this is a bacteria that loves heat. It is heat tolerant. So take a look at the temperature. The optimal temperature for this human enzyme is right about body temperature. Body temperature is 37 degrees Celsius, 98.6 Fahrenheit. So notice that's when the enzyme does the best. So the rate of the reaction is along the vertical axis. The highest rate of reaction is at body temperature. The reaction plummets quickly once somebody has a high fever, and that is because the enzyme denatures with a high fever. So if you heat up a protein too much, the bonds start to break, especially the hydrogen bonds. Remember, there's hydrogen bonds in the alpha helix and the beta pleated sheets. There's hydrogen bonds numerous times. Those fall apart in high heat, and so the enzyme denatures, and so that's why you see it plummet to not working at all with a high temperature. We usually say right around 108 degrees is when those proteins start denaturing. So a fever that starts pushing 105, 106, that runs the risk of permanent damage to the enzymes. It's not anything that any doctor can fix. Some of the enzymes can come back when the temperature cools down, but some of them are permanently denatured. Symptoms, it's often brain damage, blindness, hearing loss, and then eventually total organ system failure if the enzymes are all denaturing. So high fevers are as dangerous as a stroke or a heart attack, super, super dangerous. And so we need to keep somebody's body temperature cooled down if they're experiencing a super high fever. All right, and then if you look at still on that same graph, we see that the peak efficiency for a bacteria that loves heat is a much, much higher temperature than the human enzyme is. Now let's take a look at the next graph. In the next graph, we're talking about pH. pH is down on the horizontal axis, and it's still rate of reaction on the side. The first enzyme that they're going to be talking about is pepsin. And pepsin is an enzyme that we find in our stomachs, and it helps us with initial digestion, so breaking molecules apart. And interestingly enough, you know that there is hydrochloric acid in your stomach. The pH of the stomach is right around 2. So notice that this enzyme likes, let's break it down again because you need to review this. What is pH? pH is referencing the hydrogen ion concentration. So if there's lots of hydrogen ions, then it has a low pH. So right now in the stomach, there are lots of hydrogen ions, and that's why it has a low pH. And that's the way this enzyme works best. If you start taking away those hydrogen ions, or if you start adding too many hydrogen ions, say the stomach acid becomes too acidic, or if you take Tums and you get rid of some of the hydrogen ions, then the enzyme doesn't work as well. You want it right at that pH of 2. For trypsin, now the pepsin is one that most people get because they know the stomach is acidic. What most people don't realize is that in the very opposite, your intestines are actually basic. And so the enzyme called trypsin, which is also involved in helping you absorb nutrients, requires a high pH. And a high pH means very low hydrogen ion content. So it's typical. I think, boy, 8 seems a little bit low. I was under the impression that the enzyme pH was closer to 11, but I might be wrong on that. But regardless, the pH is higher. I hope that you pay attention or did pay attention to the liver lab, because we could add that same exact pH graph here with the enzyme called catalase. Only catalase lives in the blood. And what would the pH of blood be? Well, blood is primarily water. So the pH of blood is supposed to be right around 7. So you would see that same peak and you would see that same bell curve, only it would focus on pH of 7, which would be the pH for blood. All right, and then the last thing we're going to talk about, just again, the impact of temperature. If you raise the temperature too much, the enzyme doesn't work. Oh, I forgot to say something really important. Hypothermia. Hypothermia, interestingly, does not denature the enzyme. So that means that the enzyme does not fall apart. No enzyme, no proteins fall apart if you get really cold. What the cold does, cold, remember, is how fast molecules are moving. And enzymes are entirely dependent on their substrates landing on their active sites. But if the molecules are moving really slowly, then that doesn't happen very often. So everything's right. The active site is correct. The substrate is there, but they're bumping into each other so rarely that the reaction rate goes way down as you get colder. So somebody who is hypothermic, their enzymes are just fine. They're just moving too slowly. And this is why, I'll try to say this really fast. This is why, for example, a child who drowns in ice water can sometimes be revived after quite long periods of time when an adult would have, if they drowned, they would have been pronounced dead and their heart would never restart. It has to do with the fact that a child's body mass is so small that they get cold a lot faster than an adult's body mass does. So the child's body, their metabolism slows way down super fast. Their need for oxygen slows way down super fast. And so everything is still there, ready to function. It's just moving so slowly. So then they get that child out of the ice water and they reheat them in just the right way and they restart the heart. And sometimes we can bring that child back to life even when they've been under the water for a very long period of time. Whereas you can't do that with an adult, because the adult, when they drown, even when they drown in cold water, their body mass gets cold slower. So during the time that they have drowned, their body uses up all the oxygen that's needed, and then the tissues pass away faster than their blah, blah, blah. Anyway, so it works okay. We sometimes are able to save small children from ice water drownings, but usually not adults. All right, then pH. This is giving you one example. This would be more like the trypsin, or no, this is actually like catalase. The peak is right around 7, maybe, yeah, right around 7. So that would be an example. And then the last one, which we do look at in the liver lab also, is enzyme concentration. So the more concentrated, there are more of the enzymes that are present, the faster the reaction occurs, up to a point, of course. Once you have so many enzymes, then they don't speed up the reaction anymore because there's not enough substrate. There's not enough hydrogen peroxide, or there's not enough of the enzyme of the molecule that they're trying to break down. There's not enough of that. So the effective concentration increases up to a point and then it levels off. All right. There was one more thing. Thought we were done. I'm sorry. Okay, last thing, you guys, feedback. I think feedback is a little bit tricky. Let's see if we can talk you through feedback. So many metabolites, a metabolite is the product of your metabolism, have regulatory effects on enzymes that catalyze the metabolic pathways that result in the production of those metabolites. What does that mean, KJ? Okay, so in this diagram, please study it carefully. We're starting with an initial substrate that's called threonine. Threonine is fitting into the enzyme active site, and there's actually several enzymes that are involved in readjusting the bonds, breaking it down, or whatever, and eventually you end up with an end product that's called isoleucine. Isoleucine then can act as an inhibitor. So it says the active site of enzyme 1 is no longer able to catalyze the conversion of threonine to intermediate A, so the pathway is switched off. So once isoleucine has been made, it can turn the enzyme off. Well, why would we want to do that? If the cell has all the isoleucine that it needs, then that isoleucine falls off of the enzyme, and now the enzyme returns back to its original shape, and we start making more isoleucine. Let's try that again. So you start with threonine, it hits the active site, and then a whole bunch of enzymes work on it, and you produce something that's called isoleucine. Isoleucine gets made, and that's good. But isoleucine actually can inhibit it, so it can click on and stop that process from happening. But once there's no more isoleucine left, it falls off, the enzyme changes back into the active position, and we can make more isoleucine again. Crazy, all these things that can happen. All right, cuties, that is it for enzymes. There are a few questions that are always good for you to think through, but I'm not going to read them because this is pretty long. All right, you guys have a good rest of your day. Bye-bye.